Greetings to everyone. Welcome back once again to my educational channel on biology. Teacher Janet here. Here is a new video on an enzyme experiment to study the effect of pH on the rate of reaction or activity of amylase enzyme. Now, in our previous enzyme videos, we have discussed an experiment to study the effect of pH on the activity of pepsin enzyme, right? Not amylase. And then another video was on studying the effect of temperature on the activity of amylase enzyme. Okay, so this one's different because this one is to study the effect of pH, another factor, on the rate of reaction or activity of amylase enzyme. So in this experiment, we mix together the amylase solution and starch suspension and add a buffer solution at a specific pH, like pH 4 or pH 7 or pH 9. So the buffer solution will help to maintain the mixture at the particular pH. Then, after you have mixed all these substances together, stir it with a glass rod and also start the stopwatch. Then, every 30 seconds, take out a sample or a bit of this mixture and then test it with the iodine test for the presence of starch. So, when there's no more starch present, it means that the reaction is completed. And you can find out the time taken for the hydrolysis of starch to be completed. And from there, we calculate the rate of reaction of the amylase enzyme at different pH. Let's get started. Now, there are three experiments in this channel. on the enzymes amylase and pepsin. The one that we have discussed is studying effect of temperature on the activity of amylase enzyme. Another one was studying effect of pH on the activity of pepsin enzyme or enzyme pepsin. So this one is studying the effect of pH on the rate of reaction of amylase enzyme. Okay, pH on amylase enzyme. Now there is a very good video to watch for this experiment or practical, and I will also show you the link below so you can click on it to see the experiment. It's very simple and it can be done within about 10 to 15 minutes. All right, so let's get started. Now, so in this experiment to study the effect of pH on the activity or rate of reaction of amylase enzyme, we need to know the equation for this enzyme reaction or enzymatic reaction, which is as follows. Starch plus water with the help of amylase produces maltose. Okay, so looking at the right hand side, this is the setup for this experiment. We mix together the amylase and starch and also a buffer solution, say buffer solution at pH 7. Then at the same time, we immerse this test tube in a water bath at a temperature of 37 degrees Celsius, which is the optimum temperature for the enzyme to function, okay, at the highest rate. Now, then every 30 seconds, we take out a bit of this mixture and drop it onto the iodine solution to test for the presence of starch, right? So at first, you may get a blue-black color, right? So this shows that the starch is still present, okay? But maybe after that, you may get the brownish yellow color, which is the color of the iodine, the original color of the iodine. That means the iodine remains brownish yellow. Now, this means that the starch is no longer present in the mixture and the hydrolysis of starch is completed. Okay, so looking at this flow chart here, if starch is present in the mixture, when you mix it with iodine, the iodine will turn blue-black. So if iodine turns blue-black, it means that the hydrolysis of starch is not completed. But when the starch has all been converted to maltose and you mix the maltose with the iodine, then the iodine will remain brownish yellow, as seen here, right? So if iodine remains brownish yellow, that means there's no more starch in the mixture and the hydrolysis of starch is completed. So from there, we are going to find out and record the time taken for the hydrolysis of starch to be completed. All right, that means we have to find out the timer from this point here to this point here, okay, which is 30 plus 30 plus 30 seconds, and then plus one more 30 seconds. So it's 2 minutes. Then record the time taken, which is T in minutes, and then calculate the rate of reaction, which is 1 over T per minute. So let's discuss the details for this experiment. Activity or experiment, studying the effect of pH on the activity of amylase enzyme. So effect of something, that something is the MV, the manipulated variable. Okay. Now for other syllabuses, it's called the independent variable. All right. But here we'll call it the manipulated variable, which is the variable that you can change the values. Okay, in which you can change the values. Now on the activity of amylase enzyme, so that is your main responding variable as found in the objective. That's the main responding variable, or other syllabuses may call it the dependent variable. Okay, so we'll use the term MV, manipulated variable, and RV, responding variable here. Now, problem statement should include your MV again and your main RV, but it is not a statement. It's more like a question with a question mark at the end. So problem statement is, what is the effect of pH on the rate of reaction or reaction rate of amylase enzyme? So the problem statement is just asking the aim in the form of a question. Okay, asking the question, the aim itself. Now, hypothesis. Hypothesis should have MV and mean RV again and the right relationship. Okay, so pH 7 is the optimum pH for the highest rate of reaction of amylase enzyme, or the highest reaction rate of amylase enzyme. Here your MV is the pH, okay, and the RV is the rate of reaction. The relationship is that pH 7 is the optimum pH, highest rate of reaction. All right. Now we must also mention the other two pH used, which is pH 4 and pH 9. So at these two pH, pH 4 and pH 9, the rate of activity of the amylase decreases. Okay, now let's go on to the variables. The manipulated variable is the pH, okay, studying the effect of something, that something is the manipulated variable, or MV. The responding variable, as we said here, is either the activity of amylase enzyme, but activity cannot be measured. So to measure the activity, we measure the rate of reaction, okay, or the reaction rate. So responding variable is the reaction rate of amylase enzyme, or the rate of reaction of amylase enzyme. Okay, that is your main RV. But in the lab, how are you going to measure or calculate the rate of reaction? So we must find the time taken for the hydrolysis of starch to be completed, right, by using the iodine test, or another way of putting it is time taken for the iodine to remain brownish yellow, okay, as mentioned here. So these are both sub-RV, okay, sub-responding variable, because they are measured in the lab. This is the data that you get from your experiment. So it's the sub-RV. And from sub-RV, you will calculate the main RV, which is the reaction rate of amylase enzyme, okay, the 1 over T. Now, fixed variable or constant variable, there are many. So one good one is concentration of amylase solution, which is 1% here, or concentration of starch suspension, which is also at 1%. Temperature must also be fixed. Firstly, we can carry out this experiment at room temperature also, like somewhere between 20 to 30 degrees Celsius, without using water bath. But if the experiment calls for you to set up a water bath, then you have to, right? So for water bath, what is the temperature used? It must be the optimum temperature, which is 37 degrees Celsius. Okay, by mixing some warm water with the distilled water at normal temperature, you can get 37 degrees Celsius. Okay, the water bath to carry out the experiment. Materials: 1% starch suspension, 1% amylase enzyme solution, iodine solution, and buffer solutions of pH 4, 7, and 9. For apparatus: beaker, test tube, syringes, droppers, glass rod, white groove tile, thermometer, Bunsen burner, tripod stand, wire gauze. So these four, thermometer, Bunsen burner, tripod stand, and wire gauze, they are for setting up it's used to set up the water bath. Test tube, rack, stopwatch, marker pen, and labels. So here are some of the apparatus and materials used. The rack with test tubes in it, the syringes and the dropper, stopwatch. Here is your water bath with the beaker, wire gauze, Bunsen burner, and tripod stand, thermometer, and then a glass rod to stir the mixture of the starch and amylase. Okay, and then a white tile where you put in the iodine drops. Okay, so for solutions you need iodine, 1% starch, 1% amylase, and the buffer solutions of pH 4, 7, and 9. So what is a buffer solution? A buffer solution is a solution which resists changes in pH. When small quantities of acids or alkalis are added to it, okay, it can still maintain the pH. So a buffer solution is used to keep the pH constant. In this experiment, we will use buffer solutions of pH 4, 7, and 9, which are provided for us. So let's look at the procedure for this experiment, studying the effect of pH on the rate of reaction of amylase enzyme. So number 1, firstly prepare the white tiles here by adding 2 drops of iodine solution to each groove. Okay, so the iodine solution is brownish yellow at the beginning. Next, prepare 3 test tubes and label them as pH 4, pH 7, and pH 9. So use the sticker, the label that can stick, and then you use a marker pen to write down pH 4, and then on another label it's pH 7, another one is pH 9. Then stick separately on the 3 test tubes. Next, add 2 ml of the corresponding buffer solution to each test tube by using a syringe. So for example, for the test tube with the label pH 4, you add the 2 ml of the buffer solution at pH 4 to that test tube. Okay, and so forth. So now you've got ready all the buffer solutions in the test tube. You put 3 ml of 1% amylase solution into the test tube with pH 7 by using a syringe. Alright, so this is your FP. The volume, which is 3 ml, and the concentration of the amylase solution, which is 1%. They are fixed variables or constant variables. So add the enzyme amylase to the buffer solution in the test tube. Now, in a separate test tube, put 3 ml of 1% starch suspension, okay, into a separate test tube by using a syringe. Okay. Again, your FV is here. The volume of the starch suspension is 3 ml, and the concentration of the starch suspension is 1%. Okay, these are all FVs. Now we put the 2 test tubes, number 4 and 5, in a water bath separately. Don't mix them yet. At 37 degrees Celsius for 5 minutes for the test tubes to reach the optimum temperature of 37 degrees Celsius. So you need a bit of time for the test tubes to warm up to 37 degrees Celsius. Now, when you are ready to start the experiment, you make sure that you have tried out your stopwatch and you put it at 0. Then add the 3 ml of 1% starch suspension to the test tube with pH 7. Right? So here's your MV, pH 7, and you can now add the 3 ml of 1% starch suspension to the test tube containing the buffer solution and the amylase enzyme. That means add number 4 and 5. Add number 5 to number 4. Okay, number 5 to number 4. Next, start the stopwatch immediately and leave it on throughout the experiment. Then you do the iodine test. So every 30 seconds, a drop of the mixture from the test tube is dropped onto a new groove of iodine solution on the tile using a dropper. And the precaution is rinse the dropper well after every use so there are no traces of starch in the dropper. Alright, or else it will affect the results of the experiment. So rinse the dropper well, make sure it's clean. Now, another precaution is we must stir the mixture. Alright, stir the mixture when you mix in the starch with the amylase enzyme. Stir the mixture in the test tube well. Number 10, repeat step 9, okay, until the iodine solution first remains brownish yellow when mixed with the mixture or sample from the test tube. So if the iodine solution remains brownish yellow, didn't turn blue-black, means that there's no more starch in the mixture and the reaction is complete. Okay, this is the brownish yellow color. Alright, so step 9 is an iodine test. So keep repeating until you get a situation or condition where the iodine solution first remains brownish yellow. Then you can stop your experiment. Record the time taken for the iodine solution to first remain brownish yellow, not blue-black. Now this can be your sub-RV, or the next phrase here. This is the time taken to complete the hydrolysis of starch T. So this can also be the sub-RV, either one. Read the question carefully and follow the question, okay, in the exam. So don't assume that it's going to be either this or this. Just follow the instructions in the exam paper, okay? So we must adapt to the exam paper, right? Now, next, steps 4, where you talk about the enzyme, amylase, to steps 11 are repeated for buffer solutions at page 4 and page 9. So these are the MV, right? So you notice that all the variables are written in red here, are typed in red. Okay, all the variables are in red. Right, next. Record your results. That means the time taken for the hydrolysis of starch to be completed, T minutes, in a table. And calculate the rate of reaction of amylase as 1 per T per minute. Okay, so this is the table of results. In the first column, it's the pH of the buffer solution, 4, 7, and 9. Okay, the second column, so the first column is your MV, and the second column is the sub-RV, which is the time taken for the hydrolysis of starch to be completed, T, right, in minutes. And then your third column, the third column is for the main RV, which involves calculations. So find the rate of reaction, 1 over T, and the unit is per minutes. Okay, why is it 1 over T for rate of reaction? Because as the time decreases, the time taken for the hydrolysis of starch to be completed decreases. That means as the time becomes shorter, the rate of reaction is actually higher or higher. Okay, that means the reaction occurs more quickly. Thus, there is an inverse relationship. Okay, the rate of reaction is inversely proportional to the time. Thus, the rate of reaction is 1 over T. So here's a diagram to help you visualize what are the steps of this experiment, to help you remember the steps well. Now, firstly, I'll start with number 2 here. Prepare the tile for the iodine test by dropping 2 drops of iodine into each of the grooves here. Then, number 2, you have to prepare the amylase enzyme solution, the buffer solution, and the starch suspension. Okay, so add 3 ml of amylase solution and 2 ml of buffer solution into a test tube by using the 2 different syringes. Okay, here the volume of the enzyme and buffer solution, and even for the starch suspension, this is just an example. In each experiment, it may be different. Okay, it may vary with the instructions given. All right. So prepare also 3 ml of starch suspension by using a syringe to add this starch suspension into another test tube. Then put the 2 test tubes into a water bath at 37 degrees Celsius. Right? So wait 5 minutes to let the temperature reach 37 degrees Celsius for these 2 test tubes. So the green test tube has the buffer solution and the amylase solution. And then this orange test tube here has the starch suspension. Next, you can start the experiment. All right. So get ready your stopwatch. So start the experiment by pouring the starch suspension into the amylase solution at pH 7. Then stir the mixture well, right? And then start the stopwatch at the same time. Okay. Now carry out the iodine test on the mixture every 30 seconds. So drop a sample of the mixture into the tile which has the iodine. Okay. For example, here we see that for the first 30 seconds, when you add the mixture to the iodine solution, it turned blue-black. Let's say it turned blue-black. This shows you that the hydrolysis of starch is not complete yet, because there's still some starch in the mixture. So here is an overall picture, how to determine the time taken for the complete hydrolysis of starch by amylase. So in the mixture here, you have 3 ml amylase, you have the amylase enzyme, the starch as the substrate, and the buffer solution. Okay, now maybe it's at pH 4. Let's say it's at pH 4. Sorry, pH 7. Let's say We start with pH 7. So every 30 seconds, add the mixture to the iodine. Okay, add this mixture to the iodine drop in the groove here on the white tile. So here's an example. For pH 7, time taken for the hydrolysis of starch to be completed is how long? Now, you look at the results here. This is the first 30 seconds, okay, the first result. Then next, after 30 more seconds, and thirdly, after 30 more seconds. Now, at the second stage here, you can see there's still some bluish coloration, so the reaction is not complete until you find that the iodine solution remains brownish yellow as for the original ones, okay, original color of iodine. So example here, for pH 7, let's say this is at pH 7, time taken for the hydrolysis of starch to be completed is, now you must count the blue one, the blue spot, and also the partially blue spot, and then finally 1, the first time that the iodine solution remains brownish yellow. So it should be 3, right? So 30 seconds times 3 equals to 1 minute 30 seconds. Okay, it's actually 90 seconds, then divide by 60, you get 1 minute 30 seconds, and that's equivalent to 1.5 minutes. Okay, it's the 30 seconds divided by 60, to convert to minutes. So 1.5 minutes. Or an easier method is you just count from here. This is at 30 seconds. So the second result is at 1 minute. And the third result is at 1.5 minutes. Okay, so until this point here. So we have finished discussing the method of carrying out this experiment, plus the variables and so forth. Now let's try a biology paper 3 question on this experiment so that we can revise our technique of answering questions and prepare ourselves for the paper 3 to score as many marks as possible for paper 3. So here's a sample practical question which should be 15 marks. But for this video, I've added in more questions so for you to practice answering. So it will exceed 15 marks, right? So the more questions you try, the better. Questions on aspects like hypothesis, observation, inference, and so forth, right? So use the first 5 minutes to check your apparatus and plan your experiment. Now, let's look at the list of apparatus and materials. 1% starch suspension, 1% amylase enzyme solution, white towel with grooves, test tube rag, 5 ml syringes, stopwatch, marker pen, labels, thermometer, iodine solution, buffer solution at pH 4, buffer solution at pH 7, and buffer solution at pH 9. So these will keep the pH constant, but sometimes instead you may be given the alternative, like for buffer solution at pH 4, alternative is dilute hydrochloric acid, right, at a specific pH. Buffer solution of 7, to replace that, maybe you may be given distilled water, which is neutral also, at pH 7, right? Then buffer solution at pH 9, the alternative is dilute sodium hydroxide solution at a specific pH. So use the first 5 minutes to check the apparatus and plan the material. Now, if a water bath is used, right, you will include the beaker to contain the water, then there will be a wire gauze, a tripod stand, and a Bunsen burner, and also thermometer. Okay, but water bath may be optional as the enzymatic reaction can proceed even at room temperature. So here I've given you a set of results so that you can practice determining the time taken for the hydrolysis of starch to be completed for pH 4, pH 7, and pH 9. So try and determine the time taken for the hydrolysis of starch to be completed without looking at the answers first, right? Then we'll discuss afterwards. So pause the video. Right, so now let's look at the time taken for the hydrolysis of starch to be completed for pH 4. So if we go in this direction, this is the first result, okay? After 30 seconds. The next one is after 1 minute, right? 30 seconds plus 30 seconds, 1 minute. Then 1 minute 30 seconds, 2 minutes, 2 minutes 30 seconds, 3 minutes. Now at 3 minutes, you can see that there's still some bluish coloration. This means that there's still a little bit of starch that has not been digested or hydrolyzed yet. Okay, but in the next group here, okay, this is result number 7. If you go this way, 1, 2, 3, and 4, 5, 6, 7. Now here, the iodine remains brownish yellow for the first time, so the reaction is completed. All right. So this means that there are 7 results occurring until the iodine remains brownish yellow. So time taken for the hydrolysis of starch to be completed. There are 2 ways of doing it. Firstly, you can count the number of tiles on the number of grooves involved, the number of results involved here. Okay, until here. So it's 7, right? So it can be 7, 30. Each one occurs at 30 seconds, right? So 30 seconds times 7 equals to 210 seconds. And then to convert to minutes, you have to divide by 60 minutes. By 60. Okay, 60 seconds. Then you convert to minutes. Okay, divide by 60 seconds to convert to minutes. You get 3.5 minutes. An easy way in this case would be to just count for yourself. 30 seconds, 1 minute. 1 minute, 30 seconds, 2 minutes. 2 minutes, 30 seconds, 3 minutes. 3 minutes, 30 seconds, right? Or 3.5 minutes. Okay, 30 seconds equals to 0.5 minutes. Right, for pH 7, again, let's circle up here. You will involve only 2 results. Okay, at the second result here, around 1 minute, you can see that the solution, the iodine solution now remains brownish yellow. So the reaction is completed. So time taken for the hydrolysis of starch to be completed will be 30 seconds plus another 30 seconds is 1 minute, or 30 times 2 equals to 60 seconds divided by 60 to convert to minutes. So it's 1.0 minutes. Take note that I write to 1 decimal place, right, for every one of the results. So you must be consistent. The first results until 1 decimal place, then the second one is also okay, the same. Now, for the third set of results, what is the answer for the time taken for hydrolysis of starch to be completed? So again, we circle up all those results that are relevant, okay, until this point here, the first time that the iodine solution remains brownish yellow. So 1, 2, 3, 4, 5. That is 30 times 5, 150 seconds, then divide by 60 to convert to minutes. It's 2.5 minutes. An easier method is just to count 30 seconds, 1 minute. 1 minute, 30 seconds, 2 minutes. So 2 minutes, 30 seconds here, or 2.5 minutes. So let's look at the first question here. Now, this is the table of results, first of all. The pH of the buffer solution, 4, 7, and 9, and the time taken for the hydrolysis of starch to be completed, T, in minutes, is 3.5, 1.0, and 2.5, according to our results. Now, rate of reaction 1 over T will be calculated later. So B1, state the observation for test tube with pH 9. So it's this test tube, right? So remember, observation must have the MV, the sub-RV here, okay, which is the results, and then the reading plus units. Try and write your first observation. Try to write the observation first. Okay, let's look at the answer. When the pH of the buffer solution is 9, the time taken for the hydrolysis of starch to be completed is 2.5 minutes. Okay, so when the pH of the buffer solution is 9, this is your MV. Then comes your sub-RV, the time taken for the hydrolysis of starch to be completed. Now, copy exactly the same from the table here, okay, or from the instructions. So don't just write the time taken is 2.5 minutes. You must write it out in full. The time taken for the hydrolysis of starch to be completed. Okay, your sub-RV. So this is sub-RV. And then comes the reading, which is 2.5 minutes. So reading plus the units. Now question B2. State your inference for B1, which is about the buffer solution at pH 9. Okay, and then sometimes it's stated as explain your observation in B1. Okay, so inference is actually the explanation for the observation. Okay, so let's look at this question and what are the components of your inference. So you start off by stating whether the value here, the time, is long or short. Okay, and after that you have to relate it to the rate of reaction, which is your main RV. So from sub RV to mean RV, and how does the MV affect the RV? Okay, so we start off by mentioning the time, whether it's long or short. Now at pH 9, the time taken for the hydrolysis of starch to be completed is longer, right, compared to the pH 7. Why? So we have to give a biological reason first, because the change in pH from the optimal condition, which is pH 7, changes the charges on the active sites of the enzyme and substrate surfaces. That means the charges on the active site may be at first positive and substrate surfaces have a negative charge. Okay, as an example. But after you change the pH to 9, maybe the charges have been changed. And this caused the two parts, the active sites on the enzyme and the substrate surfaces, to instead of being attracted, they may repel each other. Okay, like having like charges, then they repel each other. So the enzyme substrate complex cannot be formed. All right, and then you relate it to the rate of reaction. So the effect of MV is stated here because at pH 9 there's the MV. The change in pH changes the charges on the active sites of the enzyme and substrate surfaces, and the enzyme substrate complex cannot be formed. So next we must mention the effect on the main RV. So the rate of reaction decreases, all right, or is lower. That's why the time taken for the hydrolysis of starch to be completed is longer. Okay, so your inference is the explanation, the biological explanation of how the MV affects the RV, the time, and then what happened to the main RV after that. Now, how about if you were asked to give the inference for pH 7, where the time taken for hydrolysis of starch is shorter, right? It's 1 minute. All right, try and write your inference here first, and then we'll discuss the explanation after this. Okay, let's see. So you have to mention the effect of the pH on the active sites and the substrate surfaces. Now at pH 7, so always start up with the timer here, whether it's short or long and so forth. Now at pH 7, the time taken for the hydrolysis of starch to be completed is the shortest. Why? Because the charges on the active sites of the enzyme and substrate surfaces complement each other. For example, maybe the charges on the active site is positive, right? And then the charges on the substrate surfaces may be negative. So they complement each other, and they will attract each other, so that the enzyme substrate complex is formed quickly or easily, causing the reaction to occur, and therefore the rate of reaction is high or low? It's high. Okay, the rate is high. And that's why the time taken is short. C. State the hypothesis for this experiment. Again, recall the components of the hypothesis. All right, as we know, hypothesis must have MV/RV and correct relationship. All right, and for this hypothesis, we use the main RV. That's the better one. The main RV from the aim or objective or the problem statement. Right? Okay, try and write your own hypothesis first. So determine your MV and main RV. Right, in this case, we don't use the format as MV increases, decreases, RV increases, decreases. All right, it's not suitable here. It can be used for temperature if the factor is temperature. Now, so for the few items, we have 3 pH used. We can compare them, right? So of all the MVs, a specific MV1 has the highest or lowest RV compared to the others. Okay, Now, in this case, for pH, we use the term optimum. So, at pH 7, pH 7 is the optimum pH for the highest rate of reaction of the enzyme amylase, right? Now, let's analyze where are our MV, RV, and relationship, okay? Now, the MV is here, the pH. The RV is the rate of reaction of amylase. And your relationship is that the pH 7 is the optimum pH, okay? So pH 7 is the optimum pH for the highest rate of reaction of amylase. Now also mention the other two pH, which is pH 4 and 9. At pH 4 and 9, the rate of reaction or activity of amylase decreases. Again, you have the MV, which is the pH. The RV is the rate of reaction or activity of amylase. And then better to use rate of reaction in this case, if it's already mentioned in the table. And then your relationship is 4 and 9 decreases, the rate decreases. So in the exam, you'll be tested on the variables. You'll be asked to state the MV, RV, or FV, and also maybe the method to handle the variables. So maybe not all the variables will be tested, but one or two of them. So state the variables in this experiment and the method to handle them. You can try out this part first by filling in your own table, and then we'll look at the answers. Okay. So what is the manipulated variable? It is the pH of the buffer solution. Okay. And then method to handle the variable. So our keywords are use something of different something. Okay, that different something is the MV. So use buffer solutions of different pH, such as, now you can give the examples, pH 4, pH 7, and pH 9. Write in the examples, right? So use something or use different something. Okay. Next, for your responding variable, what is the responding variable that you're going to write? Are you going to write the main RV or sub RV? So my recommendation is write the sub RV, okay? Because sub RV is easier to explain. So the responding variable is the time taken for the hydrolysis of starch to be completed, or it could be the time taken when the iodine solution first remains brownish yellow. Depends on the question. So read the question carefully and underline the RV there, the sub RV there, okay? Now, how are you going to explain the method to handle the variable? What are the keywords? Yes, measure and record, or weigh and record, okay? The action and then record also. So measure and record the time taken for the hydrolysis of starch to be completed, and then you must add by using something, using an apparatus. So by using a stopwatch, okay? Measure and record RV by using a stopwatch. And make sure that your RV is written in full. Don't just say measure and record the time taken by using a stopwatch. Okay, you will lose the mark there, 1 mark there. Measure and record the time taken for the hydrolysis of starch to be completed. The whole RV must be written completely. Next, your fixed variable. Okay, and you also must have the apparatus. Make sure you have the apparatus for the RV. Next, the fixed variable. What is it? So there are a few fixed variables: temperature of water bath and concentration of amylase solution. Okay, now let's look at the method to write what to write in the method to handle the variable. Fix the FV at a particular value. Okay, so fix the temperature of the water bath at 37 degrees Celsius. Or simple one is concentration of amylase solution. Fix the concentration of amylase solution at, must always have the value, fix at that value. So fix the FV at a particular value. Okay, so fix the concentration of amylase solution at 1% for all experiments. Now let's discuss the table of results. So you may be asked to draw a table to show your results and then do some calculations. Okay, draw a table to tabulate your results and calculate the rate of reaction 1 per T in per minutes. Your table should have the following titles: pH of buffer solution, time taken for hydrolysis of starch to be completed, that is T, and the rate of reaction 1 per T in per minutes. This means that your time taken should be in minutes. Right? So try and draw the table yourself first, because practice makes perfect. The more you practice, the more confident you are when it comes to the real exam. Right, let's discuss the answer. So In the first column, the first title is pH of buffer solution, that is 4, 7, and 9. For the second column, is the sub RV, right? It's the time taken for the hydrolysis of starch to be completed, T. And since the rate of reaction is per minute, that means your T must be in minutes. Okay? Now, you have to follow exactly the title that is given to you. Don't just write time taken or time taken for hydrolysis of starch. Follow until the end, to be completed, T. Okay? And then rate of reaction, follow from here, 1 over T in per minutes. Okay? Right, next. For the time taken, you can record down and write it down here. And for rate of reaction, you need to calculate. For example, for pH 4 is 1 per 3.5. Okay, show your working. It's best to show your working. And then look at the final results. The values here must be to the same decimal place, consistent to the same decimal place. In this case, we use 2 decimal places so that we can see the difference more clearly between the different rates of reaction, right? So all must be to 2 decimal places. The values must be to 2 decimal places. Right, let's see how it's marked. Now, the first one are all the titles plus the units, this way. Okay, so 1 mark for this, titles and units. So make sure you have the units, minutes here and per minutes here, right? The second mark is for your data, which you get from the experiment, your results from the experiment, okay? And the third column is for the rate of reaction, which is your calculation. So values must be to the same decimal place and always show your working to be on the safe side. So 1 mark each for every tick. This comes to 3 marks. Could be 2 marks or 3 marks. Right, now we come to the question on the drawing of a graph. So for graph A, now using the data in the table, draw a graph of the rate of reaction of amylase against the pH of the buffer solution. Okay, so in your table, actually you're using the values here, the first column and the third column here. So you can take out a piece of graph paper and try to draw the graph yourself first, as practice makes perfect. Then you compare with the graph in the answer. Right, now have you drawn your graph? Let's look at the answer. So this is the graph of the rate of reaction of amylase against the pH of the buffer solution. Right, so for the vertical axis, we write down at the top here, rate of reaction of amylase. Just copy exactly from the question and then put in the units, which is per minute. Right, without the units, then the graph is 1 mark will be deducted. Right, and then for the horizontal axis is pH of buffer solution. No units here. Right, next. Now for the horizontal axis, we don't start with 0 and then put in all the values until 4, because then the scale is too small and the graph will be very small. It will be compressed in the center, right? So we have this scale break, which is the zigzag line, okay, to indicate that some values are not shown here, and then start with 4 here. Now from this part onwards, your scale must be uniform, okay? So 4, 5, 6, 7, 8, 9. I use 3 big boxes to represent the 1 value in the pH, okay? Now, then for the vertical scale, try to use a large scale so that almost 3/4 of the graph paper space is used up, all right? So this is the scale for the vertical axis. Now, to determine the scale for vertical axis, we have to look at the readings here for rate of reaction. The highest value is 1.00. Okay, so we have to plot until 1.00 here. Yeah, just put 1.0. Now, next, for the points, okay, so you transfer the points or plot the points correctly. Okay, for pH 4, the reading is 0.29, so somewhere here. And then for pH 7 is 1, okay, 1.00. And then lastly, for pH 9, it is 0.40. So it's here. Okay, next, to use a pencil with dark or black lead, and you draw the line using your hand, okay? Draw it steadily to form a smooth curve here. So do not use a ruler to join the points, okay? Draw it freehand. Next, make sure that you do not extrapolate or go beyond the points, the first point and the last point. Okay, so you must start with the first point and draw the graph, and then after that end at the last point, but don't extend beyond the last point. All right, so you should get a smooth curve. You should draw a smooth curve, okay, to get the mark for curve. So to repeat, if you look at the points here, write the correct titles with units for each axis. Okay, the title is units rate of reaction of amylase, and then bracket the units is per minute, right? pH of buffer solution. Now follow from the question here, pH of buffer solution. Exactly follow the titles given here, right? Then mark the points correctly with the symbol X, right, on the graph. Join all the points freehand to form a smooth curve. Then fourthly, do not draw a line of best fit as for some sciences. So you must join all the points and don't miss any point. And do not extrapolate beyond the endpoints. Okay, so that's all for the graph, for this graph. Now, how about bar chart? Okay, let's say a question is using the data in the table, draw a bar chart, not graph, bar chart of the rate of reaction of amylase against the pH of the buffer solution. So your two titles are still the same, but now you are drawing a bar chart. It's different. Try and draw the bar chart based on the data here, okay? And then we will discuss of this. All right, let's look at the answer. Now, the bar chart consists of the rectangular boxes, and there are no curves, no lines in this case, right? So you can start at this point here with pH 4, and then pH 7 and pH 9. So what is important here is that your bar must be of even the same thickness, okay? And then again, you have the titles pH of buffer solution, the rate of reaction of amylase in minutes per minute against the buffer solution, right? So, and then you must plot the bar so that the uppermost part of the bar is at the reading is the same as here, okay, in your table of results. So for pH 4 is 0.29, for pH 7 is 1, and for pH 9 it is 0.4, right? Then the distance between this bar and this bar must be the same. And don't stick the bars together. For bar chart, you don't stick the bars together unless it is a continuous variation where you get a normal distribution curve, okay? But usually the bars don't stick together. Don't stick the bars together, okay? Or you may lose 1 mark for that. So mention the 3 pH here, pH 4, pH 7, and pH 9. So the marking could be 1 mark for the correct transference of the points, and 1 mark for the shape of the bars. They must be of equal width, right? And then 1 mark for the titles and the scales here. Now let's look at a different graph, graph B. So using the data in the table, draw a graph of the time taken for the hydrolysis of starch to be completed against the pH of the buffer solution, right? So in this case, you are not asked to draw a graph of the rate of reaction of amylase against the pH, but instead a graph of the time here taken for the hydrolysis of starch to be completed. All right. So how would you draw this graph? So take out a graph paper and practice drawing a graph that involves these two sets of data, right? Okay, let's look at the graph now. So for this graph, for the vertical axis, it is the time taken for the hydrolysis of starch to be completed, and that's in minutes. And for the horizontal axis, the title is pH of the buffer solution. Okay, next, again for our pH, our values are 4, 5, 6, 7, 8, 9. So we try to draw a bigger graph, so we have to stretch out, make a bigger scale, choose a bigger scale. And also for the vertical axis, the time is from 0 till 3.5. That is the largest value. So I try to use the maximum space in the graph on the graph paper. That's why my scale is like that. Three large boxes here is for 0.5 units or 0.5 minutes. Okay. So anyway, because the reading is 3.5, 1.0, and 2.5. So all are like halfway, 2.5 and 3.5 can be easily determined here. Right, so after I have drawn the scale and written down the titles plus units, I just mark the values. Okay, for pH 4, we have 3.5 minutes, right? And then 7 is 1.0 minutes, and 9 is 2.5 minutes. So you can see here that the graph is different already. It's the reverse of the graph for rate of reaction against pH. Right now it dips downwards. In the previous graph, the graph goes upwards and then comes down. Okay, this one goes downwards and then comes up like that. Okay, so be careful about the RV that you're asked to use in the graph. Depends on what RV is. Is it the sub RV or the main RV? All right, so this is another different graph compared to graph A just now. Next question, based on the results of this experiment, define operationally the hydrolysis of starch. 3 marks. So for operational definition, which can be asked even though it's not so frequently asked, please take note of the components, right? And then try to write your own operational definition. Right, have you done so? Let's have a look at the answer. Now, in your operational definition, you try to put in the theoretical definition, but modified to the practical. State the organism or substance used. In this case, the enzyme, amylase, and starch. You must mention these two, right? RV. So you must write is shown or determined by the particular RV. And then CV, you can add in, if not, never mind. Okay, but you want to make it complete, can add in. MV, for MV, we say that the RV is affected by the main MV. So mention what the main MV is. Okay, which is the pH. So in other words, it's DORCAM, DORCAM. Think of DORCAM. Now C here is for constant variable or fixed variable. Now if you don't add the C, that is DORAM. So some answers don't have the C, the constant variable. Okay, based on the results, define operationally the hydrolysis of starch, right? Or sometimes it can be asked, give the operational definition for the hydrolysis of starch. So hydrolysis of starch, start with this term that you are being asked. The hydrolysis of starch. Now, what's the meaning of the word hydrolysis in the textbook definition? It's the breakdown, right? Breakdown of starch. So here we add in the organism of the substance by the enzyme amylase into maltose, right? And then the RV, it is shown or determined by the time taken for the iodine solution to first remain brownish yellow. Okay, because in this case, you cannot say it's shown or determined by the time taken for the hydrolysis of starch to be completed. Not so clear. So time taken for the iodine solution to first remain brownish yellow. So the hydrolysis of starch is, now here's where we use affected by. Affected by the MV, the pH of the reaction medium or water bath, okay, or the pH of the pH of the buffer solution. So this is the last question. The experiment is repeated using a buffer solution at pH 7, but in a water bath at 15 degrees Celsius. Predict the results of this experiment and also explain your answer. So there are two parts, predict and then explain. Now, for this second experiment here, is using buffer solution at pH 7, right? But what is changed? The temperature, okay? So in the original experiment, the temperature is 37 degrees Celsius. Now you're using water bath at temperature of 15 degrees Celsius. So is it higher or lower? It's a lower temperature, okay? So you should be able to predict the results of the experiment. That means mention whether the time taken for the hydrolysis of starch to be completed is longer or shorter, right? Now you can try to answer this question first and then give an explanation for your answer. Right, let's look at the results. P is for prediction. The time taken for the hydrolysis of starch to be completed, or time taken when the iodine solution first remains brownish yellow, will be longer than 1.5 minutes. Why 1.5 minutes? Because 1.5 minutes is the time taken for the hydrolysis of starch to be completed at pH 7, but in water bath at 37 degrees Celsius. Okay, so you have to refer back to your table of results and find out what is the reading for buffer solution at pH 7, 37 degrees Celsius. That's 1.5 minutes. So now at lower temperature, the time taken for the hydrolysis of starch to be completed will be longer. Okay, so explain why. So E1, this is because the enzyme amylase becomes less active at lower temperatures. So there are less collisions with the substrate and less reactions occur, right? So the reaction rate is lower. Okay, it's lower. So mention what happens to the rate of reaction. This is your main RV. So for prediction, always mention the main RV. What happens to it? The rate of reaction of amylase decreases at lower temperatures. Okay, so notice that the first explanation relates to the biological reason, okay, of the effect of temperature on enzyme amylase. And then the next one is to state what happens to the main RV. Okay, right, that's all for this lesson. I hope you have benefited from it and learned or sharpen your techniques of answering the paper 3 questions. So all the best to you for your paper 3 and goodbye for now.